Dr. Joanna Bloese is the entomology extension specialist and is based in Hilo. Her research focuses on plant-insect interactions, specifically for integrated pest management strategies. She is also examining the applied effects of silicon-soluble fertilizers as a strategy to reduce pest damage. She strives to bring creative problem-solving perspective to her research and pest management tactics. Dr. Bloese will be presenting Integrated Pest Management Research Updates. Dr. Bloese, thank you for joining us today.
Hi, Glenn. Hi, everyone. Thank you for that introduction. HFNA, thank you for the invitation. It's a pleasure to be here. So, as Glenn mentioned, I'm going to give updates on the silicon research that I've been doing over the last year. This is in collaboration with Joanna Imamura and Russell Galanti. And really what I'm going to talk about is its potential for resistance management strategy for various thrips in Hawaii ornamentals. So with that, I will share my screen. As I mentioned, we're going to go over some of the management tactics for thrips. And if you're an ornamental grower here, you probably have issues with thrips. I know it's everybody's favorite insect, and there's tons of different species here in Hawaii, especially a wide assortment that affect anthuriums and orchids. So just to name a few to refresh your memory for those who are less familiar, from left to right, we have chili thrips, which are a big pest of both anthuriums and orchids, banana rust thrips, melon thrips, which is the top right picture here. And these pictures, a lot of these are from Arnold Hara, my predecessor, who did a lot of work in the management of this insect. And then we have Western flower thrips. Both melon thrips or Thrips palmi and Western flower are big invasive species here in Hawaii. And if that wasn't enough, the list continues. Other thrip pests of these crops include orchid thrips, anthurium thrips, and banded greenhouse thrips, which you see on the right from left to right. And this list isn't even exhausted. So if that's 7 thrips already, and we still have onion thrips or Hawaiian flower thrips that are issues for growers here as well. And thrips really cause damage through their rasping, piercing, sucking mouth parts. They can cause damage to many different parts of the plant, to the leaves, the stems, and most importantly, to the flowers, which is our product that we're trying to sell. So let's go on to some damage. Sometimes different thrips have different types of characteristic damage we'll find in the field, and some so much so that it's a way to kind of identify them. Thrips, if you're not familiar, are very, very small. They're very, very hard, nearly impossible to identify as species in the field. So these types of techniques can help. Western flower thrips is pretty characteristic to see some browning, especially on the colored spade, like non-white anthuriums that have a spade that's colored. It's often kind of this zigzag pattern. You might have to cross your eyes to kind of notice the zigzagginess of them. For anthurium thrips, which are highly abundant in this crop, you often see this whitening or silvering on the spade. At high infestations, you'll see deformation of the spade altogether, which is what you see in picture 3 with the chili thrips, and often associated with that is heavy scarring. If you see scarring or twisting, deformation of the stem in an anthurium, you're probably looking at some melon thrips there. So let's talk about some thrip damage on orchids or dendrobium specifically. Melon thrips are a big issue for dendrobium as well as orchid thrips, and they cause scarring and discoloration on the petals and sepals, most definitely. But the biggest problem we experience from thrips in dendrobium is they really attack the new emerging spikes. They love that young growth. It's highly nutritious, has lots of nitrogen. They cue into that kind of yellow-green color of brand new growth, and they can completely deform the buds, the emergence of the spike. It can lead to very few blooms per spray, and it can greatly reduce yield. As any dendrobium grower knows, they have to spray often. So talking about our research, and I'll get into the nitty-gritty in a little bit. What did we find in our experiments? So these were greenhouse experiments in a saran house with open sides. We did not inoculate our flowers with a certain type of species of thrips or a certain count. We just had natural populations infest these plants. The most common insects we found were by far thrips. That was our biggest pest by far in the anthurium trial. And that included anthurium thrips and melon thrips and banana rust thrips. In the white variety, we can see this kind of stippling. It might be a little subtle, but in person, it's a little more easy to see. And then in the right picture, that's typical of anthurium thrips there, that streaking and that white mottling effect there. So now with the dendrobiums, let me put a quick plug. You will get to see at the field day tomorrow these plants in the field. This trial has been up since August 2020. So these plants have been in the field for over a year and they have not received any insecticide spray. We sprayed the anthuriums once with spinosad. And so they're not the happiest plants right now, but if you do nothing, you'll get to see kind of the decline of the plants versus if you just sprayed silicon. Again, I'll go further into detail in a bit, but when we look at the dendrobium trial, yes, we did see a lot of thrips damage. That picture from earlier where that spike did not form correctly. But we also saw a lot of fungal issues, which is not uncommon, especially on the helo side of Big Island. So we saw issues with fusarium, and then these are early stages, and then the first 4 pictures, and then we saw the more typical chlorotic melting look of that fusarium. The picture on the top right is botrytis, another fungal issue, and we also saw mealybugs. The majority of this was on the control treatment. Okay, so before I get into the nitty-gritty of the research itself, I wanted to quickly just put this diagram in. I think it's a really nifty diagram developed by Ken Lenhart and Kelvin Sawaki out of a publication they produced a while back about dendrobium production in Hawaii. So as a way to kind of target your monitoring strategy, where to look on the plant for various arthropod and mollusk pests. So like I had mentioned, we had issues with thrips, which we always found on the spray and the flowers themselves. They love to tuck themselves deep into that flower. And of course, the mealybugs like to hide as well between where the crevice, where the leaf kind of meets the cane. And we had some mealybug issues, and then ants. Ants will come and try to tend to those mealybugs. And they may come at the bottom of the plant, but you oftentimes see them everywhere. So getting into the experiments themselves, there were 2 projects. One was soluble silicon fertilizer on anthuriums and its effect on various, its efficacy on reducing damage from various pests, but mainly thrips. And this was an RCBD. It was a randomized complete block design with 8 reps and had 6 treatments total. The second trial is orchid, so we'll go into that next. So this is us setting up the field for this project, and this is exactly what you'll see. Well, there'll be plants in the field tomorrow, but one of the stations for tomorrow's field day will be this greenhouse in particular. Working hard, or not so much. But okay, getting into the treatments. So there were 2 varieties of anthuriums that we looked at. One was a white variety. This was developed by Dr. Tessie Amori. To my knowledge, I don't believe it has a name yet. It's a UH variety. We would have to check with her if that's still true. The other variety we used was an obaki, called Honi Honi. It was kind of this pink obaki. And the different treatment levels within each variety were control or untreated. So all of them received the conventional amount of your typical Nutricoat fertilizer. And then we did untreated, and then we did a silicon drench, and then we did a silicon drench plus foliar. And that total is 6 treatments. These are the plants in the field about 1 month into the trial. So they were getting established. We transplanted these, we top cutted. The Honi Honi and the whites were from Floriculture. And so they did experience some stress, but they all started around the same size with 3 leaves. Okay, so silicon, the product we used was from Redox. It's called Mainstay SI, and the formulation is 22% silicon dioxide and 10% calcium silicate. And we used a rate that was really high. The rate we used was 3.27 gallons per acre. And this was because I was getting advice, actually. Dr. Suzuki, John Suzuki, who spoke earlier, he got me in touch with Dr. Wendy Zellner, and they've been both advising me on this project. And she is an expert in silicon, and she's out of the University of Toledo. So she's recommended we go with this high rate. And if we go with this high rate, then we know if it's effective, great. We can do more research at lower rates to see if it's just as effective. But if we started with a lower rate and it wasn't effective, we might wonder, oh, was a higher rate effective? So we started high and went low. So when I refer to silicon fertilizer, what I'm referring to is silicon, the element on the periodic table. And this is easily confused with silica or sand or a mineral that makes up quartz crystal, for example, that has a very specific structure to it, or silicone, which is the synthetic polymer. So when I refer to SI or silicon, even if I misspeak, I apologize. I'm really referring to that element itself. And you might be asking, why are we studying this element, this fertilizer? And it's really because there was an emerging body of research that came out that really demonstrated that silicon in general is supposed to help with these plants' natural defenses, fortify cell walls, and reduce stress. Silicon is actually the second most common element in Earth's crust. However, all of that is not available in a form available to plants to uptake. So in a system like anthuriums and dendrobiums, which are typically, and not always, grown in cinder and blue rock, we could assume that it's unlikely a lot of silica is available for them for uptake. And there's very little research done on the effects of silicon fertilizer in semi-epiphytic and epiphytic plants. So that was kind of our reasoning behind why we wanted to examine this element. All right, now getting into the good stuff. Just give me 1 sec, I gotta take a sip of water. I'm not used to talking this much. Okay, so there's a lot of research or a lot of results here I'm gonna try and cover. I might talk a little bit quickly, and this isn't everything, so I'll give another update once we've done the full entire analysis. What you're seeing here, this graph, is just the white variety of anthurium, and it plots total marketable flowers over time for 10 months. And that's the amount of time total that we took data on these flowers. Well, marketable yield data, yield and quality data. And the green is untreated, the yellow is drench only, and the purple is drench plus foliar of SI. And so, there's some times when purple is above green and purple and yellow is above green, but not that often. It's not consistent. It's quite inconsistent. We don't really see a clear effect of the treatments on this white variety, which is what I had mentioned a year ago. We had thought, we're 2 months in and we're not really seeing a clear effect. But what's very interesting, and what I mentioned earlier, was that in January, the thrips pressure got so high that we had to apply a pesticide. So we applied spinosad, and we were getting, essentially for all intensive purposes, 0 marketable yield in flowers. And of course, you can see right after we applied that spinosad, it worked, it did its job. And all of the lines, all of the colors do go up in yield, but the 2 silicon were not different from each other. And that is from January to February. That's where we saw the biggest separation between the control and the silicon lines. So what does that mean? It could mean that there's this synergistic effect. And so including silicon occasionally or always with your pesticide applications, you could have a greater effect, and it could be managing thrips better. And hopefully you would have to apply pesticides less frequently. So that's something we want to look at in the future. This is a graph of just 6 months, and it's the total. So it's irrespective of time. The treatments are the same, associated with the same colors, and it's still total marketable yield. It's just a little easier for our eyes to see. So the difference between control, the foliar and drench did produce more flowers, but it's a difference of about 17. So whether that's cost beneficial with how much we applied the silicon every other week, maybe not. The rep of the silicon product said it's about $7, an additional $7 per application per acre. We applied it at a much higher rate than that. So we would have to do that cost benefit analysis to really know for sure. So now getting into the honey honey. Okay, so it's the same chart we were looking at for the whites, total marketable yield over those 10 months. This time blue is the untreated, orange is the drench only, and gray is the foliar plus drench. And this time it's nice because in August, all of the lines start relatively close together. And it's a difference of like maybe 8 total flowers out of all of the plants out in the field, which was 288. And then we see the silicon lines, orange and gray, start to really diverge within a month from the control treatment. And they stay above the blue line, which is the control, until January. And they start to get closer and closer to that blue line leading up to January. So as I mentioned before, silicon seems to be somewhat effective, but with increasing populations of thrips, it'll have a diminishing benefit in your field. So it is not a replacement for insecticides at all, right? We're just trying to maybe apply fewer pesticides per year, and maybe they can complement each other in that way. No surprise, in January, after we applied spinosad, all of the lines have an upward trajectory, which is great. So if we look at this graph again, which is irrespective of time, we're seeing a bigger difference in the honey, which is what we saw in the timeline graph that I just presented. And it's a difference of about 37 flowers, marketable flowers, between the control blue and the foliar and the drench. And the drench only is not far behind the foliar and the drench. There is, at the rate we applied, a clear white residue on the plants, which might actually be beneficial. I'll get into that a little bit later. All right, so another thing we looked at was the silicon accumulated by the plants. So about a couple months ago, we took leaf samples and we sent them to the lab, ADSC, and had them look at how much silicon was in that plant tissue. This graph, it has divided it by treatment. The obaki is the honey, and that's the pink, and the white is the blue. And the very interesting thing that I don't quite have an explanation for is the controls for both had higher SI accumulation than the SI treated plants. And not significantly so. I think the only one that was significantly so was for the obaki. Obaki control had significantly more silicon than the obaki silicon plus drench, but it was not any clear significant difference for the whites. And I find that very interesting, especially because the obaki honey were the ones where we saw the biggest effect of silicon. So I'd really have to talk to John Suzuki and Dr. Wendy Zellner again to try and understand what's going on there, the biological mechanism. As I think I mentioned earlier, I roped Russell Galante into this as if he wasn't busy enough. And he did the base life, what I was calling post-harvest data for this treatment. So after we would harvest every other week, we would give our flowers to him after we labeled them. And he had certain parameters that he monitored over time. And once they reached a certain threshold, he would discard that. And he used previous literature that he went off of and modified it for our needs, I believe. If you have any questions, you can talk to him again tomorrow at the field day about that. So I'm just going to go over this really quickly. He knows more than I do, but as you can see, there's no clear differences between the treatments, but there are clear differences between the varieties. So white variety had more spadix browning. That was the reason for discard than honey. And it's not too surprising that honey had more bluing than the whites. I don't think the whites typically blue. I think that's characteristic of anthuriums that have color in them. How am I doing for time? I think I have to speed up a little bit. A couple other parameters that he monitored were spathe browning and spathe gloss. Again, no clear trend with spathe browning, no trend among treatments or variety. With spathe gloss, no clear trend among treatments, but among variety, yes. We saw that whites were more likely to be discarded due to loss of gloss. So, and then one thing he added was stem rot. We kept finding what I call decapitated flowers. The stems got a bacterial infection or some fungal or some infection and would die or fall off. And we saw a lot more of that in the whites. by this graph. And we also saw this be the only graph where silicon reduced the number of stem rot in the white variety. So while it didn't do much in the field, in this vase life, it did seem to reduce stem rot by about 10% there. Okay, getting into the orchid. I love this trial. I find it very interesting. This is a bit of a different design. It was a split plot design, and it had 6 treatments total, and only 6 reps instead of 8. This time, instead of having 2 varieties, we have 3. Just for simplicity, I'm going to call them white, lavender, and purple. We did not tease out the SI from a drench and foliar. It's just pure SI, which every other week we switched from drench to a foliar application to a drench, so on and so on, and a control. So these plants have been in the field for 13 months now, and we have not sprayed any fungicide. We have not sprayed any pesticide. They received conventional fertilizer, and then the only thing else they received was silicon, and the controls received nothing. So the plants are hurting, but I would love for you guys to come to the field day tomorrow to see them and give me your opinion of what you think. Do you see any difference? Is it noticeable in the field, or is it just from the data that we learned this story? So this is a picture about maybe a month into the trial. And from this picture, it looks like the plants on the right are much taller, but that's just the angle of the picture. That's not actually really the case. And I'll just get right into the results. So looking at thrips damage, which was a big problem for the dendrobium here. I mean, dendrobium growers know that if you don't manage thrips, you're not going to have a crop. And that's exactly what we saw. And it was pretty amazing how quickly we saw it. It seemed like the highest thrip pressure was in the lavender. They love it. My guess is the scent. They're drawn to that scent, similar to how we probably are. And that is where we saw the biggest effect of silicon. It was absolutely amazing. So if you do nothing, you'll have for lavender about 75% reduction in yield, where if you just apply silicon, no pesticides, you'll only have a reduction in yield by about less than 20%. And it was significant irrespective of variety. And so this graph on the right is what we're looking at irrespective of variety. So it seemed to reduce thrip damage by half. Now, that doesn't mean it necessarily translates into reduced yield, right? As Russell mentioned in his talk with the virus A, if it's just alone, there didn't seem to be a lot of decline symptoms, but as soon as it's co-infected, then you do. But here we of course, and it's not surprising, we did see it translate into marketable yield differences. So with lavender treated with silicon, we had 100% marketable yield, meaning of what we harvested, what percent of that was marketable and not. So, so the number of spikes that they put out and sprays they put out, they all were able to, they might, be missing one bloom on a spray, but they were still marketable. Versus the control, very few were. And that's trend is through both varieties, all varieties. Okay, so this slide, what I think this slide shows are kind of 2 things. And this is a slide of average percent marketable yield over time, just for the purple and the white variety. And what I think it shows is that first, it takes a little bit of time for the silicon to take effect. So from November to December, there is a huge, there starts to be this huge separation between the lines, and that separation is maintained. So if you're going to use silicon, you kind of have to be patient. You got to wait for 2 to 3 months before the silicon becomes effective. What you don't see in this graph that I really want to mention is that very similar to anthuriums, when thrips pressure gets high, which it's lower in the winter months, as everyone knows, when it gets warmer, when there's less rain, we see more thrips, especially under greenhouse conditions. These are not shade house conditions. We see reduced effectiveness of silicon. That was unanimous across both experiments. And so in the summer, even though we weren't collecting data, this data goes through March. I have data through May. I have to add it. So unfortunately, we don't have data through May to August and September, which would be nice, but we see decline in silicon effectiveness. Okay, Russell also did base life for dendrobiums as well. And again, just biological data here that reduced damage translated into longer base lives and longer sprays. So, and it was very significant. And it kind of followed a similar trend about, half or double the length of the spray. The average was around 20 for control versus 40 for silicon. We also did a silicon accumulation test through the lab, ADSC, for the dendrobium as well. We took tissue samples. And the interesting thing here is that the silicon treated plants did accumulate slightly more silicon than the control plants, but none of them were significant except for the white variety. The white variety accumulated significantly more silicon. But honestly, and I'm just thinking of this now, it seems like the lavender is where the silicon was the most effective, and it had the lowest difference in silicon between the silicon and control treatments, which is similar to what we found with the anthurium. So there might be something biological or genetic going on there. So to sum up, what we know and what we don't know. For anthurium, silicon significantly reduces thrip damage on honey variety, and it reduced stem rot on the white variety, but overall, it seems to be variety dependent. Occasional applications of insecticide greatly enhanced its effectiveness, and the two together might be very beneficial. For the orchids, the beneficial effects of silicon were evident. It reduced thrips damage by half and doubled marketable yields. Something I didn't have time to talk about here, but you'll see tomorrow in the field day is that it also seemed to greatly reduce fungal infections, and it increased base life. Things that we don't know, the magnitude of the beneficial effects of applying pesticides in conjunction with silicon. And then given that, I don't know the optimal timing or frequency of insecticide applications and what that should be. And I also don't know what mechanism is the reason for this effectiveness of silicon and it reducing pest damage. So that kind of basic research might be something that's really interesting to look at. And then one disclaimer I want to say is that, this silicon is a fortifying fertilizer. And I know I'm running out of time. It's not an insecticide, but I think when we examine its effectiveness at managing insect damage, it's tempting, to want to compare it to insecticides. However, it's not a substitute for pesticides, but it may allow us to apply them less frequently and increase the lifespan of these tools that we have by slowing evolutionary resistance. And it could also likely be a physical barrier for some of these insects, so you might not get resistance as quickly. These are just some examples across the different pictures I took across the different treatment levels for the white variety of anthurium and the red or the honey. This is not always true. You had some within the silicon that were completely decimated by thrips. They were just less frequent. And then stay tuned for things that we're going to continue with this work. We're going to do a cost benefit analysis. We kind of want to know what mechanism is responsible for this observed difference. And again, examining SI in combination with other cultivation practices and other symptoms. I had a grower look at try it in Kentia. He said it was really effective. So I'm interested in doing maybe a greenhouse trial with that. And Hannah also showed interest in seeing if they were effective in Protea, maybe at a lower rate where we don't get a residue, hopefully. With that, I want to thank the following. And I could not have done this without them. Ryan Porter did the bulk of the data collection. Tiani Cow inputted the data. Joanne Litchie, my collaborator. Russell Galanti, Elton Mao, Angel Magno, Dale Tusha, Jerry Hay for the flowers, Eric Tenoy, all of these people helped greatly, and all of my sponsors as well. So with that, I will take any questions if there's time. So sorry to go over.
Thank you,
Okay.
Dr. Bloese. Yes, unfortunately, we are out of time. However, you know, your silicon research looks real exciting. You know, we look forward to updates in the future and see how it's, you know, progressing, and especially, you know, its effects on the other crops that you had mentioned. So thank you very much. Okay.
Thank you.
You're
Bye, everyone.
welcome.